Okay, in this video we are going to talk about sex linkage. This is the case where you get certain diseases, certain genetic diseases, which really only affect males and they don't affect females. And we'll see why, the reason for that in a minute. But what we've got here is called a karyotype, is a human karyotype. So a karyotype is when you get cells undergoing mitosis, you add a poison, colchicine, to stop mitosis, and take a photograph of the cells, and then you can have a look at during mitosis, of course, you can see the condensed form of the chromosomes. So the photographs taken and the homologous pairs are matched up. The old way of doing it would be to cut the photograph up with a pair of scissors and match them up. So here we can see in the human karyotype, then we've got 23 pairs of chromosomes. 1 to 22 are called the autosomes. And the last pair, the one down here, are the sex chromosomes. Now we can see this particular karyotype must have come from a male, because you've got an X chromosome, and next to it you've got a little short Y chromosome. And you see there's a region of the X chromosome which is not matched by the Y chromosome, and that's really important. We'll see in a minute. So that would be a male. All females have 2 X chromosomes. Okay, so let's see how this can affect disease then. Right, I'm going to take the example of an autosome. I've got a autosome. Let's say we've got chromosome pair number 7, chromosome 7. We've got 2 of them, 2 homologous chromosomes there, and let's think about the gene for cystic fibrosis. So F would be the healthy version of it, and F is the mutated version. And remember that F, this codes for a chloride ion channel, and the mutated version doesn't work. It won't let chloride ions through. So if this person here, let's say they've got the locus for the cystic fibrosis is up there. Let's say they've got 1 good copy there, and here we have got on the other chromosome 7, we've got little f, the mutated version. Now that person is heterozygous, so they wouldn't suffer from cystic fibrosis because they've got 1 copy of this. They're able to make a good version of the chloride channel program, so they don't have any symptoms. Let's think about if this was on chromosome, we had a mutated gene on the X chromosome then. Now, the X chromosome, right, let's say we had a female, has a gene H, which I'm going to write down there, a locus H, and this codes for clotting factor 9. Clotting factor 9, that is needed. There are lots of different proteins required for your blood to clot. So when you cut yourself, there's a chain reaction, a cascade of reactions between these clotting factors, which are proteins, which eventually lead to the formation of a clot. If you're missing out any of those factors in the cascade, then the clot won't form properly. And so when a person, they can't produce any good clotting factor, when they cut themselves, they'll bleed. Okay, and that is a disease called haemophilia. It's generally only experienced by males, and untreated, which you couldn't be treated before about 1930, the sufferers of it would die before they reached adulthood. So let's have a look here. Now this female has got 1 bad copy of the gene, small h there, but she won't have the disease because she's got 1 good copy, and that is enough to make adequate amounts of clotting factor. Let's think about a male though. Say if a male has 1 bad copy of the gene. So there's the X chromosome and there's the Y chromosome. Now the male will have, if he's got a faulty copy there, then he will suffer from the disease, because here there's no matching part on the X chromosome, no matching part on the Y to match that part of the X. So even though he's only got one bad copy of the gene, he hasn't got a good copy, so that person will suffer from haemophilia. Okay, so we're going to do see how this is inherited then. Okay, so let's think about the different genotypes and things we can have. Okay, so we put it like this. So this will be a female. So that female has got 2 X chromosomes, each one bearing a good copy of the gene. So that female would be healthy. This person would be a female. They'd be healthy, but they would be a carrier. They could pass it on to any sons that she has, but because she has got one good copy of the gene, no symptoms themselves. Then we could have a male. That male would be healthy, but this male, even though he's only got one good copy, he has got haemophilia. So let's do a cross then. Let's see what happens when we have a healthy female, but she's a carrier. And okay, now generally speaking, until recently, people with haemophilia, males with haemophilia, so if you had that genotype, they wouldn't get to reproduce because their health wouldn't be good enough, although that is changing now. But let's see if a healthy carrier with a healthy carrier with a healthy male. So the gametes that they can produce are, well, the female, she's got 50% of her eggs will have that X chromosome, 50% will be like that. The male, 50% of his sperm will have X chromosomes with a good gene on it, and 50% will be Y. The sperms will have Y chromosome, and that obviously has no gene at all for factor X. So we draw a little diagram that we usual way. So we'll have the male gametes up here, and the female gametes here. And we can see how this is going to work out. Right, so we put the genotype. So this one here, she will be a healthy female and will not be a carrier. This will be a healthy male. This one will be a healthy female, but she will be a carrier. And this one will be a male who will suffer from the disease. Okay, so let's just do that. So this one is going to be disease, healthy, this one here, healthy and not a carrier, and this one here, she will be healthy, but she will be a carrier. Interestingly, it's a very famous case in the royal families of the British royal family, who were related to the German royal family and the Russian royal family, the Tsars before they were killed in the revolution. They were all interrelated, and haemophilia was rife through those families. And it turns out Queen Victoria must have been, she had a son who had haemophilia, and she had grandsons who had haemophilia. Queen Victoria must have been that genotype. She must have been a carrier herself. She never suffered any ill effects. So let's just see, if you have a carrier female, what's the chance of her producing a son who has got the disease? Well, you can see half of her sons on average will be healthy and half will have the disease. So male offspring will be 50% healthy and 50% with the disease. Female offspring, well, they'll all be healthy. But 50% of them will be carriers. Right, now it's maybe quite interesting to talk about codominance here. And sometimes it's very a little bit oversimplified, and it will be said, right, such a gene is dominant and such is recessive, other ones are codominant. There's a spectrum really in reality. Now with the mutations for the factor 9, it's not just one mutation which causes the disease. There's actually hundreds of different mutations have been identified. And usually when a female is a carrier, they don't have any symptoms, but occasionally, depending on the particular mutation, it is possible sometimes for a female carrier to have mild haemophilia. Right, the other gene, which is well known for being X-linked, it's called X-linked, is colour-blindness. That's a little bit more complicated than clotting factors, though. If you want to research it, you can do this. It can be although it's generally speaking only males who can get colour blindness. Some females can suffer from it as well. But it is the gene coding for, it's actually 3 genes which detect colour. They're called opsins. One detects blue light, one detects red light, and one detects green light. And it's usually a mutation in either the red or the green, which causes color blindness, which means that, and those genes are all on the X chromosome, a male who has a mutation in either of those genes has difficulty in distinguishing between red and green colors.